It's not always important that you understand the very structural and functional aspect of a molecule. It's also sometimes important that you give it a name which can be understood by anyone who is dealing with the subject. So there must be some, what we say, universal rules to give a name to any compound. So that's why we say for enzyme also we have a nomenclature. That nomenclature is used to give a shorter names, easily understandable and universal names which can be understood by anyone who is dealing with the subject. So mostly the enzymes have a suffix ase, like lactase, lipase, hydrogenase, and these have with them, with the ase there is the name of the, what we say, the substance which is being acted upon by the enzyme. For instance, lactase for lactogen, then we have galactase for galactose, lactase for lactose, glucosidase is similarly an example which affect glucose, and so on and so forth. And then also sometimes with the name you get the very phenomena which is involved in the reaction which is acted upon by or which is controlled by the enzyme. So description of action is also given there. For example, lactate dehydrogenase, aldehyde acylase, isomerase, and so on and so forth. So the action is also given with the name of the enzymes. But it's not always applicable to all the namings. For instance, we have certain enzyme names which are not following these rules. They are just trivial. For example, pepsin, trypsin. I mean, these, they don't have any suffix or they don't have any action name, but still these are being used. Then there is also indexing of the enzyme for really giving them a universal name. For example, for lactate oxidoreductase, there is the indexing of 1.1.1.27. So though it makes it more universally acceptable, but at the same time it can be cumbersome also. It can be also confusing also. So one have to be careful when giving or memorizing the names of the enzymes through this indexing. Then there are 6 major classes of enzymes. I will just give you a little bit of the name. This is just for the sake of letting you know about the classification of enzymes. We are not going to in detail that how this enzyme performs this function. There is oxidoreductase, as the name shows. They are involved in the oxidation reduction reactions. There are transferases which are involved in the transfer of one group to the other group. Then there are hydrolases which are involved in the cleavage of the bonds. There are lyases which also involved in the cleavage of the bonds other than the water, I mean in which water is not involved. Then there are isomerases which tend to, I mean, rearrange the molecules so that the structural aspects of the molecules are changed, but not the molecular formula is changed. And then there are ligases which join 2 or 3 molecules. So they are general classes of enzyme or types of enzymes which you will be studying again and again throughout this course. You will be definitely dealing with many of these class of enzymes. There are some confusing nomenclatures also. Synthetase versus synthesis. Similarly, phosphatase versus phosphorylases, and similarly dehydrogenases versus oxidases and oxygenases. So they look similar in name, but they have different actions. Synthetase requires ATP, for instance, while synthase does not require ATP. Similarly, phosphatase uses water to remove phosphoryl group, while phosphorylase uses inorganic phosphate to break a bond. Similarly, dehydrogenase, in dehydrogenase, NAD and FAD act as electron acceptor, while in case of oxidase, oxygen acts as an acceptor. But here oxygen is not incorporated into a molecule, but in the case of oxygenase, the oxygen gets incorporated. So these are little bit of confusions when you are talking about the nomenclature, and you have to keep these things in mind whenever you are studying the enzyme.